Hello, Bruins, and today we're going to talk about DNA history and the structure of DNA. Um, so we started out talking about mitosis, and one time we know that when a cell divides, that the nucleus is divided too. So in the past, people had an idea that whatever was in a nucleus had to be being passed on to the cells. So the nucleus is the key in order to answer what is the hereditary material. Now, hereditary material determines genetic variation among population, and all this is due to the hereditary material. So the big question is, in the nucleus, we knew that there was protein and we had DNA. Now, the person who kind of discovered something that we call now nucleic acid was Friedrich Mischer, and he worked with he looked at pus from bandages, and he noticed this material was different from protein. It did not contain sulfur, but a lot of nitrogen, phosphorus, and it was very acidic. So he coined that term nucleic acid. But he himself kind of believed that the hereditary material must be protein, because protein was a good candidate because it had 20 different amino acids and it could have a great variety of protein. So let's kind of go through a series of experiments that helped us understand that DNA is indeed the hereditary material, and then later on we understood the structure of DNA. So in the 1920s, we start with Friedrich Griffith. Now, he was working with an experiment where he was trying to find a vaccination against pneumonia. And he worked with mice and bacteria, and he worked with the bacteria called Streptococcus pneumonia, which causes pneumonia. So there was two strains, rough strain that was harmless, and then there's a smooth strain that would cause a disease. It was virulent. So he worked with the hypothesis that if he injected the heat-killed smooth strain bacteria, which caused a disease, and the harmless R strain, then the mouse would live. So basically his first thing was, of course, his control, inject the mouse with, so this one here is the smooth, the R strain, right? So this one, the R strain, the mouse lived. And when he injected the smooth strain, the mouse died. And then the heat-killed S strain, the mouse lived. So then his hypothesis control group was, this was his question, that if you took the heat-killed S strain and combined it with the R strain, then the mouse should live. And that's what he expected. But did he find that? No. So what happened is, when he did inject the heat-killed S strain and the R strain, the mouse died. And he couldn't understand why. So what he did was he took a blood sample and he looked at it, and he noticed a bacteria in the dead mouse was found to be transformed into the deadly S strain. So this led to him conclude that there's some kind of transforming agent in the S bacteria that caused our bacteria to change, and this transforming agent must be the hereditary material. And it's most likely DNA, not protein, because protein are very sensitive to heat. So if he had heated it, he should have had destroyed the S strain. So the next experiment was Avery, McCarthy, and McCleod in the 1940s. What they did was they took Griffith's experiment and used enzymes. And the two enzymes they used is protease, and this is an enzyme that would break apart protein, and they used DNase, an enzyme that would break up DNA. And to determine whether it was protein or DNA, the transforming factor. So here's their experimental setup. So they took the heat-killed the heat-killed S bacteria and the R strain, and they added protein, the protease enzyme, and the protease enzyme would destroy the proteins. And when they injected, the mouse died. So this rules out that it's not protein, because here the proteins are dysfunctional. Now here he added DNA, so this destroyed the DNA, and when they injected into the mouse lived. So that means it must be DNA. So they conclude the transforming agent is DNA, not protein. Then we have another experiment, and this time we're looking at viruses. And everyone knew that viruses infects the host. It injects some kind of hereditary material that would take over the host. So here Hershey and Chase experimented with bacteria that viruses that infects bacteria called bacteriophages. So here's a picture of a bacteriophage, and basically bacteriophages are basically protein and hereditary material, DNA. Now what they did was they know that protein contains carbon, oxygen, hydrogen, and nitrogen, and sulfur, and DNA contains carbon, hydrogen, oxygen, nitrogen, and phosphorus. Okay, so if they can tag what's different between the two, then they can trace what is being injected into the cell. So they tag the sulfur with the radioactive material, and they tag the phosphorus. So here's their experimental setup. They're going to infect the bacteria, and the first batch is radioactive protein, and then they blend it to shake off the viruses once it infects the cell, and then they centrifuge it. Now whatever is in the liquid part, that's what we call supernatant, and that's going to be whatever is outside of the cell, and whatever is in the cell is pellet. So and then they checked for radioactivity, and they found that the supernatant was radioactive, so that ruled out it's not protein that was injected into the cell. Now they did the same thing, and this time they tagged the DNA, and when they allowed the virus to infect the bacteria, and they shook off the viruses off the cell, centrifuged it, and checked for radioactivity, and they saw that the pellet was hot or radioactive, then they concluded that it is DNA is the hereditary material. Okay. Now, just a little bit about viruses. Viruses infects the host by first attaching to the host. So basically it's like a protein tail that would attach, and then the hereditary material, DNA, or it could also be RNA, is injected into the cell, and that takes over the host DNA, which makes the host DNA make viral parts. And once the viral parts are assembled, the viruses are ready, the cell would burst and more viruses would be released. So this is something what we call the lytic cycle. In other cycle, the viruses could just incorporate its DNA or RNA into the host genome and be dormant, and that would we call lysogenic cycle. Now, other experiments that later on that contributed was about DNA was, one example is Shirgoff. Now Shirgoff looked at different animals and noticed that they all had equal proportions of adenine and thymine and cytosine and guanine. And then the next big experiment was done by Wilkinson Franklin using X-ray crystallography. And this is the famous photo 51, that Rosalind Franklin took. And she worked with X-ray crystallography. And X-ray crystallography is basically taking the reflection of the DNA, and basically it shows that that the DNA has a helical structure. So Watson and Crick saw that picture and took Shirgoff's idea and Rosalind Franklin's famous photo 51, and her assumption that the backbone must be composed of sugar and phosphate, and the rungs are the bases. And when Watson and Crick put the model together, they figured that the DNA, the backbone, is anti-parallel, and that the bases were adenine and thymine paired up, and guanine and cytosine paired up. And so with that, with Shoggoth's idea, and they created a model, and the model just fit right in. So here is the DNA model. So you have the backbone of DNA. You have here the phosphate group, the sugar, and notice the sugar is attached to the base. So this is what we call a nucleotide. And this is one strand of the backbone of the ladder, and here's the other one. And notice they're anti-parallel. So this sugar and this sugar is like upside down. And then here, what we call the rungs of the ladder is our bases. And notice the G always pairs up with the C, and the T always pairs up with the A. And they're bounded together by hydrogen bonds. So we always kind of refer to DNA to a helical, a double helix, because it's double-stranded. Now RNA, on the other hand, is a single helix. Okay. Now, another important thing about is when we learn about replication is, here's your DNA nucleotide, and our first carbon is attached to the base, and here's our second carbon, and our third carbon is what attaches to the next phosphate of the next nucleotide, and here's our fourth carbon and our fifth carbon. So when we look at our DNA, if you notice the fifth carbon, so that's why we call this 5'. And here, the last carbon that ends with is the third carbon, so that's why we call this end the 3'. And then here, our 3' is on top, so we call this strand 3'. And here, the fifth carbon is the last one, so we call this 5'. So we're gonna next go into replication, and that'll be my next screencast.